Okay, class, so today we're going to cover membrane proteins and translocation, and then towards the end we'll look at some post-translational modifications that happen in the endoplasmic reticulum. The first part of today's lecture should be review on membrane proteins. You guys should all have this. And as you know, membrane proteins are involved in a wide variety of functions, right? Not only attachment, but also cell signaling and some other things like that. And most notable is the ability of transmembrane proteins to transduce signals across biological membranes, right? Or move ions or molecules across the lipid bilayer. Now, membrane proteins require protein sorting and localization. Almost all membrane proteins in the cell are actually made on the endoplasmic reticulum, excluding some proteins in the mitochondria and stuff like that. And the communication across a membrane typically involves transmembrane proteins. And basically, membrane proteins are either embedded in the membrane or they can actually sort of be associated with other proteins that are membrane bound, or they can actually acquire a lipid anchor. So here's some examples. Okay, so typically a membrane protein will span the lipid bilayer. Okay, so here in this instance you have an alpha helix spanning the lipid bilayer, but you can also have a lipid anchor holding it there. You can have multiple transmembranes passing to create a transmembrane protein, and we've sort of seen this with G protein coupled receptors that have 7 transmembrane domains. You can also have proteins that create sort of pores using beta barrel formations, and then you can have shorter alpha helices that are just really hydrophobic, and they can basically insert themselves into half of the lipid bilayer leaflet. Okay, you can then also have lipid anchors either on the cytosolic side or the cytoplasmic side. We'll look at a couple of these. Certain G proteins will have a lipid anchor, and then proteins on the outside can have this thing called a GPI anchor. And then you can get peripherally associated membrane proteins that are associated with an integral membrane protein. Okay, and it can either be on the cytosolic side or on the the outside of the cell. Here's just some examples of some lipid anchors that can happen. You can have an amide linkage between the amino group and amyristic acid. Sorry, I have trouble pronouncing some of these, which basically creates a myristoyl anchor. You can have a thioester linkage between the cysteine and palmitic acid or palmitic group, and then you get palmitoyl anchor, and then a thioether linkage between a cysteine and a prenyl group to generate farnesyl. And here's what these things sort of look like in the chemical structure. You guys don't really need to memorize these. I think it's just really important for you guys to understand that there are lipid anchors that can be attached to proteins post-translational, and then these can actually then sort of anchor the protein into the lipid bilayer. As it pertains to membrane-spanning alpha helices, these are typically on the order of 20 amino acids, sometimes a little bit longer, and they're just very, very hydrophobic. Okay, and so you'll have a stretch of 20 or so hydrophobic amino acids, and this will create a perfect alpha helix, and this alpha helices will then want to be embedded in the membrane. Okay, now any individual sort of membrane-bound alpha helix, depending on the amino acids contained within it, can actually self-associate with other membrane helices and then displace the lipids, and we'll see that. And there's actually computer algorithms that allow you to predict the hydrophobicity or the likelihood that you're going to have one of these sort of hydrophobic transmembrane alpha helices, which can then sort of predict whether or not you're working with a membrane protein. And you do this by this technique called creating hydrophobicity plots. You can just enter the amino acid of your sequence into a computer program online, and it basically gives it a hydrophobicity index or a hydropathy index here. And then if you see that you have a stretch of 20 or more amino acids that create sort of a very hydrophobic region, this is highly likely that this is going to be a transmembrane protein. Here's actually bacterial rhodopsin, which we know is in the G protein coupled receptor family, and you can see the 7 transmembrane domains that sort of exist on it. Okay, you can do this throughout the whole entire genome and see how many membrane proteins a given organism will produce, and typically it runs around 18 to 20%. So here you can see all of the proteins that are produced by E. coli, and a fraction of them are membrane proteins. Here you can see it in S. cerevisiae, or better known as the yeast we've been looking at, and then as well as in Homo sapiens, or man. Okay, so roughly around 20%. And these all represent sort of these membrane spanning domains, so you can predict how many membrane proteins are actually encoded in the genome. Now, you don't need a full alpha helix to form a membrane domain, and so there's, exceptions to every rule. But here in the case with aquaporin, you have these 20 amino acid or so membrane spanning alpha helices, but then there's also these split helices that are roughly probably about 10 amino acids, and they come together in such a way as to almost form an entire alpha helices, except they're sort of split. The great thing about membrane proteins and the alpha helices is they tend to self-associate, and oftentimes you can then, still exhibit its function. So, for example, if you have a single chain multi-pass membrane proteins and you cut it with a protease, it's still going to form the protein based on the hydrophobic and hydrophilic packing of the alpha helices in here. And you can also then take it and randomly cut it and express 2 versions, and it will also come together to form this. Okay, so there's this whole thing with transmembrane self-assembly of transmembrane proteins based on the amino acid sequence of the alpha helices. And you can sort of view that as, this particular newly synthesized membrane proteins with its hydrophobic alpha helices, then transmembrane alpha helices, that'll actually come together to create a folded protein, and in the process displace the lipids that are there. You can also have beta barrel proteins, and these exist in microbes like E. coli, and here's just basically the periplasm and the extracellular space, and you can see these beta barrels of OMP-A. Outer membrane protein A is what OMP stands for, and then you can have a 12-stranded OMP-A, OMP-LA, 16-stranded porin, or a 22-stranded FEP-A. And when you have these sort of beta barrel configurations like this, they can actually make great pores, and hence the name porin, right? It forms this great pore. Okay, one of the things that happens to proteins in the secretory pathway is they get post-translationally modified. Okay, and the post-translational modifications are different depending on the side of the membrane that it's on. Okay, so you have your cytosolic side, which is actually a reducing environment. If you guys think back to, I think it was lecture 2 or 3, where we were talking about oxidizing environments and reducing environments. Well, the cytosol is a reducing environment, so you're going to have free sulfhydryls in the cytosol. However, in the lumen of the ER and on the extracellular side of the plasma membrane, you're going to have these basically intrachain disulfide bonds where you get the disulfide formation. And these disulfide bonds are actually formed in the ER and by an enzyme called protein disulfide isomerase. Another thing that happens or starts in the endoplasmic reticulum and then happens and gets modified further as it goes through the Golgi, as we'll see when we're covering vesicular transport, is that secreted proteins get the addition of oligosaccharides, or better known as basically glycosylation. Okay, so glycosylation starts in the endoplasmic reticulum, and then when you get to the Golgi apparatus, it gets further modified. And you can also have interchain disulfide bonds where you're basically creating a protein out of a bunch of different polypeptides. And a perfect example of this is the antibody, which contains, which is basically a single protein composed of 4 polypeptides, 2 heavy chains and 2 light chains that are connected by these disulfide bonds. Now, this glycosylation that you hear, these oligosaccharides, that actually creates a carbohydrate layer on the plasma membrane of the cell, on the extracellular side of on the extracellular surface of a cell. Okay, and so here, if you stain the carbohydrates, you can see the extent of protein glycosylation that's on the outside of the plasma membrane. Here's the cytosol, and here's the nucleus. Okay. And if you wanted to sort of look at that in sort of a schematic format, what you end up having on basically the exterior of a cell are all of these glycosylated proteins as well as glycolipids that are there, and that actually creates that carbohydrate layer on the cell. And so here you can just see different proteins and their different glycosylations schemes, and you can also have these things called proteoglycans, which are heavily glycosylated proteins, and these are all very common in the extracellular matrix. Okay, so studying membrane proteins actually creates a bit of a problem, because you have this lipid bilayer and it's not very soluble, so it's hard to separate these proteins. And so from cytosolic proteins, which are just soluble in water. So if you want to solubilize transmembrane proteins, what you have to do is you have to add detergents. And there's a whole class of detergents out there. Here's just 3 examples that we use in the lab quite regularly. Okay, and these detergents then help solubilize the membrane proteins by basically disrupting the hydrophobic interactions that occur. And there's 2 main types of detergents. There's basically ionic versus non-ionic, and the difference is the charge. Okay, so sodium dodecyl sulfate here, SDS, which is a common compound used in a lot of buffers we use in the lab all the time. It actually has a charged negative sulfate group and a sodium ion associated with it. Another one that's non-ionic is this one called Tritinex, which basically has no charge on it. And one of the things that you need to know about detergents, like lipids, they're amphiphilic. Okay, now in order to solubilize membranes, you actually have to get to this point, which is called the critical micelle concentration. And so the critical micelle concentration is basically defined as the concentration of detergent that will basically create micelles. Okay, and so anywhere below the CMC, the detergents basically are existing sort of as monomers. But then when you get to the critical micelle concentration, you actually start to create these micelles, and that's basically what the CMC is. Okay, so I bet you're asking, what is a micelle? Okay, well, so if you sort of had a schematic of amphiphilic detergents here, where you have a hydrophilic head group and a hydrophobic tail, when these start to sort of coagulate and form a spherical molecule like this that's soluble in water, what you end up having is what's called a micelle. Okay, and so here it is. And these detergent micelles, if they're in the sort of the high enough concentration, instead of existing as monomers, they can actually start to solubilize membranes. And here's how that works. So here's just a typical lipid bilayer with a membrane protein. And then, you have basically your detergents, and some will exist as monomers, and others will exist as detergent micelles, and they're sort of in equilibrium. Oftentimes, if you're above the CMC, you're more in this state right here. And then when you basically add the detergents to the membrane protein, what you're going to have is the detergents coating the hydrophobic portion of the membrane protein, as well as sort of rounding around the corner and solubilizing pieces of membrane. Okay, and this is basically how you solubilize the membranes. And what you can do is all kinds of cool stuff with this. You can not only purify whatever protein you want, but then you can reconstitute liposomes or whatever. And so to sort of give you an example of that, here is basically your lipid bilayer with your transmembrane proteins. And let's say you wanted to purify this sodium-potassium pump to study how it works. You would basically add in detergents above the CMC, or the critical micelle concentration, that would solubilize the membrane proteins and basically the lipid detergent micelles. Then you can go on and purify your sodium-potassium pump, and then if you want, you can remove the detergent and sort of reconstitute the lipid bilayer with the sodium-potassium pump in it, and then you can do some functional studies on that protein. Okay, so it's just a sort of a neat technique, and this whole thing about CMCs, anyone that's planning on going to graduate school, or those of you in graduate school, this just becomes a really important concept, that and knowing the difference between an ionic detergent and a non-ionic detergent. And one of the things I didn't say about the ionic detergents, which I want to talk about, is an ionic detergent will not only disrupt hydrophobic interactions, but it will also disrupt electrostatic interactions. Okay, and so in that instance, that's also a denaturing condition. So whenever you add in an ionic detergent, it's considered under denaturing conditions, whereas if you add a non-ionic detergent, it's considered under native conditions. Important concept, especially for the graduate students to appreciate. Okay, so now let's start looking at a few transmembrane proteins. So there's bacterial rhodopsin, which once again is in the family of the G protein coupled receptors. It has 7 transmembrane spanning domains. It sort of coalesces as a patch on purple bacteria, and here you can sort of see a patch of it on the cell. If you do an EM, you can see that sort of coalesced even closer together and more tightly packed. Then if you look at a single bacterial rhodopsin molecule, here's what it looks like, and these things can come together. Okay, and what bacterial rhodopsin is it's a light-activated proton pump, and so the bacterial rhodopsin will actually absorb a photon of light, and in so doing it will move a proton in sort of this numbered path. Okay, so the proton jumps to an amino acid, and then it's sort of pumped out into the extracellular space, and then basically a proton is pulled off another lysine, and then another proton is pulled up from the thing. So it basically pumps this proton across the membrane. Another light-activated protein pump is the photosynthetic reaction center. Okay, this basically is found in photosynthetic bacteria, and we'll look at this more when we're looking at photosynthesis. We'll also look at the photosystems in plants while we're doing this. But in general, what happens is you have these central chlorophylls right here, sort of shaded in green, and they'll absorb a photon of light that excites an electron down, and eventually travels down these pigments and eventually ends up on a quinone. Okay, and Johann Diesenhofer and Hermann Michel actually solved the crystal structure of this back in the late 1980s, and they won a Nobel Prize for it. And this is actually the structure of it. Okay, and they defined how the electrons moved and how it works as a proton pump. Okay. Now, we sort of covered this last time when we were looking at the black membranes, but as well as some computer models, but you actually have lateral diffusion of membrane proteins, right? Just like you have lateral diffusion of the lipids in the lipid bilayer, you have lateral diffusion of proteins. And that's pretty easy to see. You can take a mouse cell and a human cell and you can sort of force them to fuse in the laboratory. Then if you're using fluorescently labeled antibodies to mouse proteins and fluorescently labeled antibodies to human proteins, you can add those in, and eventually over time they're going to distribute equally across the entire cell. Okay. You can also watch lateral diffusion of membrane proteins by doing this experiment called FRAP, where you take a membrane protein and you fuse it to like GFP, and that will be in the membrane, and then you photo bleach it. So this is FRAP stands for fluorescence recovery after photo bleaching. So you can take an area and you can photo bleach it, in this case this little square, and then over time there's going to be sort of lateral diffusion and it will recover, and you can sort of measure that by the bleaching and then the recovery in that spot if you're just measuring from where the square is. Okay, so that's basically the FRAP technique. You can also do the opposite called FLIP, which is fluorescent loss in photo bleaching, where you basically have GFP fused to a protein, a plasma membrane protein, and then you can sort of continually bleach a specific area and measure in an adjacent area, and after continuous bleaching it's going to basically bleach out all of the GFP signal till eventually you have a cell that's not producing any color to it or any fluorescence. Okay, so you can see it sort of fall off over time as you continually bleach a specific area. Now these are just techniques to measure the dynamics of membrane proteins on the plasma membrane, and in individual cells they can tend to just laterally diffuse unobstructed. However, cells in the body actually do have obstructions which prevent their lateral diffusion, which is oftentimes a good thing because that gives you the ability to create polarized epithelial cells. Okay, so in this instance you have a polarized epithelial cell where you have sort of an apical surface with an apical plasma membrane protein shown in green. Then you have these tight junctions that don't allow the proteins any lateral diffusion to the other domain, and then in the other domain, the basolateral domain, you have lateral plasma membranes and basal plasma membrane proteins. Okay, and these oftentimes will sort of connect to the basal lamina. And ultimately, these sort of tight junctions basically restrict the diffusion of plasma membranes and create a polarized state. You can see that in actually a lot of cell types where cells create sort of membrane subdomains. And so if you look at here, you have basically a guinea pig sperm, you have the anterior head, the posterior head, and then the tail. And these all represent different domains on the sperm, and you can label different proteins. So here's the anterior head, the posterior head, and the tail domain. You can also have these membrane subdomains on the cell itself, right? And we sort of saw this with the bacterial rhodopsin where they were sort of coalesced together into small groups. But then you can also have scaffolds within the cell that are holding transmembrane proteins in a specific region. Or if there's a contact outside the cell, the plasma membrane proteins can associate with that and hold them in place. And as you guys have seen, you know, when you have any kind of synaptic junction, the proteins will actually coalesce. Okay, so you've got basically an immunological synapse or neuronal synapse. The proteins that are needed will oftentimes attach to one another. And these aren't mutually exclusive. You know, in the case with neurological synapses, you'll oftentimes have scaffold proteins underneath holding the proteins in place. So it would be a combination here of C and D. Okay, you can also have things in proteins that, you know, or plasma membrane proteins and stuff like that, that help maintain a specific shape of a cell. Okay, and so that can then be attached to the cytoskeleton and can actually restrict the diffusion. Okay, and so here's a perfect example of that with red blood cells where they sort of create these disc like properties which allow them to travel through the bloodstream a lot easier, okay, without getting clogged. And what does that are these proteins called spectrin and ankyrin, where you have a spectrin dimer connected to actin, and then you can have other proteins associated like tropomyosin, and yeah, and then ankyrin proteins here shown in yellow. Okay, and this will help sort of hold everything together and help give the cell its cell shape, right? And in doing this, it also limits the lateral diffusion of proteins like this glycophorin that's here as well. And here's just an EM of the spectrin and ankyrin as it's sort of attached to the surface of a red blood cell. And if you sort of follow that, you can just sort of draw it out schematically, where you're going to have these basically these membrane skeleton proteins, that are attached to the membrane, and then you'll have your transmembrane protein. And these sort of membrane subdomains have the ability to limit lateral diffusion. So if you were to follow this transmembrane protein over time and look at the various domains, and you were to plot it out, it sort of starts here, it's in one domain for a while, sort of stuck. It can occasionally escape, and then it's in this blue domain for a while, and it's sort of stuck just going around, and then it can enter other domains and sort of travel around. Okay, and so ultimately, these membrane skeleton proteins and the associated proteins that are there, like you see with spectrin and ankyrin in red blood cells, these sort of create barriers. They're occasionally passable barriers, but they're barriers that inhibit diffusion. Okay, so that's all I have for part one of this. We'll do part two in the second, where we're going to look at translocation, and we'll begin to look at transport through the secretory pathway. Okay, thanks.